Hello, everybody. Thank you so much for joining us. Our speaker today is Sara Roccabianca. She is an associate professor at Michigan State University, where she runs a lab focused on experimental and theoretical modeling for biomechanics research. She has done work on multiple soft tissues, and today she's going to be sharing her work on the mechanics of the left ventricle. So thank you so much, Sara, for joining us today. And do you want to do questions during your talk, after your talk? Any preferences or?
I don't have any preference, but if we're doing during the talk, I may need a moderator to help me out because with the Zoom, sometimes it's hard to keep track. So if
Okay.
you see someone raising their hand, stop me anytime. I'm fine with that.
Perfect. Okay, so if anyone wants to interrupt with questions, do a hand raise or put it in the chat and I'll draw attention to it. All right, go ahead.
Okay, thank you so much for the introduction and for inviting me. This is very exciting. I haven't seen virtually so many people in so long. So it's really cool to be here. And today I'll talk to you a little bit about the existence of interconstituent mechanical interaction between myocytes and collagen in the left ventricle. And I like to start this talk with a little bit of a guessing game. And these are between these four pictures, two represents soft tissues and two represents human-made composite materials. And I'll give you the answer in just a minute, but I think first it's interesting to just focus on how similar they look when you look really close. These are all SEM pictures. And so here's the answer. These two represent the myocardium, and these two are human-made fiber reinforced materials. And this is just to show that it's just a justification of why we think we can use homogenization theory and material homogenization theory when we describe soft biological tissues. And just a couple of things about constrained mixture and homogenization theory. So when we look at composite materials through this theory, what we do is we evaluate the behavior of the overall material. Can you see my pointer? Okay. We describe the behavior of the overall material by a weighted average of the behavior of each of the constituents of the composite. And in this case, it's weighted by the volume, and sometimes we weight it by the mass. But anyway, each material contributes with their own properties and their amount. And this is what the general idea of this theory. But there's a couple of notes here. And one of them is that when we look at this type of homogenization, we make the hypothesis that the materials cannot, they can only contribute by their presence, and they cannot push and pull on one another. And another hypothesis we usually make is that when the composite is unloaded, every constituent within the composite is unloaded. And we know that these two bottom hypotheses may not be true or are generally not true for soft biological tissues. For example, we know that when you look at soft tissues really up close, you can see this is a schematic of the microstructure of the myocardium. You can see that myocytes and collagen fibers are really touching one another physically. And so it's reasonable to believe that during the contraction, but also in a passive condition, these two materials can be exerting mechanical forces on one another. And we also know that in soft tissues, no loads applied does not mean unloaded material. In fact, we are all familiar with the existence of residual stresses, which one can see, for example, applying cuts to the tissue that can release this existence of these hidden internal forces within the soft tissue. And so really the idea behind of this study was to try to understand what is the mechanical environment that the cell really feel, and if this mechanical environment can be affected by the interaction with surrounding constituents such as extracellular matrix, and in this case, collagen fibers. And so we think it is crucial to be able to estimate what is the actual mechanical environment around cells, and this is important when we're thinking about mechanotransduction and mechanobiology. And so, and then the next question was, do we think it's important to account for this interconstituent interaction if it exists in a modeling of a soft tissues? Because when we're looking at growth remodeling models, for example, a lot of these are based on estimating what is the stress state, and especially the homeostatic stress state within cells that cells respond to. And the growth is based on variation of this homeostatic stress state. And so it becomes crucial to understand exactly what is the stress within the cells, and if it is affected by the interaction with other constituents around it. And so, as I said, this study focused on the left ventricle, and you can see here, this is the same schematic that I showed before. Down here, you can see an SEM picture that we took of the native myocardium. This is a rat from a Sprague-Dawley rat. And when we're looking at the composition of the left ventricle has two main constituents, or at least the two main that we are considering in this model. One is the myocytes occupies the largest part of the volume in the left ventricle, and it's really the workhorse of the heart, and it's the one that makes your heart beat. And then the second constituent is the collagen fiber network that, despite occupying much less of the volume of the left ventricle, is still thought to contribute strongly to the mechanical behavior of the tissue. And so we're trying to understand and investigate how these two constituents interact and how the interaction can affect the tissue mechanics. So the specific questions we were trying to answer were how myocytes and collagen fibers both contribute to the mechanics of the left ventricle, and also how the mechanical interaction between myocytes and collagen fibers what is the role that it plays in the mechanics of the left ventricle. We decided to try to answer these two questions using two main approaches, one experimental, by performing a series of opening angle experiments and investigating the residual stresses in the left ventricle, and also by using a series of manipulation of the tissue and tissue treatments to isolate, not just study the intact native tissue, but also to study the isolated constituents, both collagen and myocytes. And then we paired this series of experiments with a mechanical model, which we call CMMI, which is a bit cheeky, but which is it's a continuum tissue level constrained mixture model that can account for the mechanical interaction between collagen and myocytes. And we use this to interpret, and the experiments are both used in the sense of informing the model, as well as we use the model to try interpret some of the experimental results we obtain. So I've been told to focus on the methods. So there's some methods included in this talk today. And so first, I start with the tissue preparation. So all the results that I'm going to show you have been collected from hearts of adult Sprague-Dawley rats. And after we have excised the hearts, both males, and we have studied both males and females. After excising the heart, we have cut two lateral rings, and you can see here an example of a representative ring from the left ventricle. You can see here the left ventricle, and on this side is the right ventricle over here. After isolating these rings, we have tested all the intact tissue, which contains both myocytes and collagen, but also we have performed decellularization to isolate collagen fibers, and we have also applied a collagenase treatment to the tissue to isolate, to the best of our ability, the myocytes. You can see here, these are three SEM images that we collected to show the intact tissue. And then here on the left is the isolated collagen after the decellularization process. And here on the right, it's the isolated myocytes after the collagenase process. So a couple of details about tissue preparation. So when we looked at the intact tissue, we soaked the tissue in a modified Krebs buffer, which contains BDM, which is a myosin inhibitor, because we were trying to prevent as best as possible contraction of the myocytes. Then when we were performing our protocol to isolate collagen fibers through a decellularization, what we did was to soak the tissue for 96 hours in a STS solution, which is an ionic detergent that has the ability to disrupt biological membrane and lysate the cells and remove them from the tissue. And the 96 hours time point has been evaluated through a pilot study in our lab. And then finally, when we have isolated the myocytes, we have performed a collagenase treatment, and for which the tissue has been first soaked for 50 minutes in the Krebs buffer plus PDMN, and then we've added both calcium and bacterial collagenase. The addition of calcium was necessary to activate the enzymatic degradation by the part of the collagenase. The tissue was treated in this solution for 96 hours, and then it was put back into Krebs plus PDMN to try to prevent contraction of the myocytes again until testing. All of these protocols have been informed by literature review and then optimized in our lab through a series of pilot studies. After this, we have performed a series of imaging studies to try to understand how good our methods to separate the constituents were. And so you can see here a series of histological studies. So on the top, you can see three microscopy pictures of H&E stains, and at the bottom, these are polarized light images of a picrocereus red stain. We also performed Masson trichrome stain. And you can see here in the center, there's all constituents. And here on the left, you can see that in the H&E image of the isolated collagen, there's a largely decreased presence of myocytes, and this gave us confidence that our decellularization process was actually isolating the collagen. And on the bottom here in the polarized light, you can see that while the fibers here, it's kind of hard to see a little bit, but here on the right, you can see the fibers are mostly green, which suggests if not a complete absence of the collagen, a strong decrease in the diameter and degradation of the collagen, which we think decreases the contribution of the fibers to the mechanical behavior. While here, both in the isolated collagen and in the intact tissue, you can see the fibers are mostly glowing red and yellow, which suggests more stronger and fully formed fibers. We've also applied a MATLAB quantification, like we have an in-house MATLAB code that we use to quantify constituents in histological pictures, and we have seen that in the control samples, we have a distribution of a 10% collagen and 90% myocytes presence. But after collagenase treatment, we see that the collagen decreases to 3%. And after decellularization, we see that the presence of the myocytes decreases to 11%. So this is the quantification we did to prove our treatments. These are the same SEM images, which we also did to try to figure out if we did the treatment in an effective way. Then we performed the experiment, the opening angle experiment. Here you can see from the native ring, this is intact tissue pictures, we apply a radial cut, and then we use this graphic procedure, which is adapted from this paper of Omens and Fung in the 90s. We use this graphic procedure to estimate the opening angle. You can see here the detail, and what I want you to focus on is that we took 10 for each sample, we took 10 pictures over a time of 90 minutes. So here's some results after the opening angle experiment. You can see here at the bottom, there's the time after the cut and on the horizontal axis, and on the vertical axis, the value of the opening angle that's been measured. We have here both the male in blue and female in pink, which is, cheeky again, results. And at the bottom here, you can see represented with squares the opening angle for isolated myocytes. In the center here with diamonds, intact tissue, and at the top with triangles, isolated collagen. First, we tried to figure out if these results give us reasonable results. So we only had comparison in literature for intact tissues of male, but we have seen that the value we recorded of about 58 degrees after 90 minutes was consistent and comparables to what prior studies. And we've also performed some statistical analysis and comparison with the results represented in this paper from Rodriguez and colleagues, and we found that there's no statistically significant difference between our results and theirs, which gave us confidence that this test gives reasonable results. And for the female, we believe this is the first time that this result has ever been reported, so we didn't have any results in literature that we could compare. And then when we compare the three different treatments, we saw that there are statistical differences at every step of the way through the test between the three groups for both males and females. And this is also consistent with what has been seen in arteries before, where degradation of collagen reduces the opening angle, which is what we observe here from the intact tissue through the myocytes. So we see that the myocytes have the lowest contribution to the, as the lowest value of residual stresses, and the collagen has the highest value of the residual stresses, while the intact tissues assumes a value in between. So these are the experimental results, which now we're moving into more of the modeling that we have employed to describe and interpret these results. So this is the constitutive description we have used. Up here, again, there is this homogenization that we're doing when we're looking at the intact tissue, which is weighted by their volume contribution. Here is written in terms of volume, but what we're doing is really a area examination through histological samples. But this is the relative contribution, relative presence, within the tissue. Here it's how we evaluate the Cauchy stress, and here you can see here the two strain energy functions that we have adopted for this study. And to evaluate the mechanical parameters for both isolated myocytes and isolated collagen, what we have done has been to look into the literature and find experimental measurements of mechanical behavior of isolated myocytes cells and isolated collagen. And we have reproduced those results from the literature, and then we've used our mechanical model to estimate the material parameters, as you can see here. And then what's the innovation of this study is the kinematic. Here's a general view of what the kinematic of our model is. And I'm gonna describe step by step each one of these mapping that we're doing. And I will start from these two independent evaluation of the residual stretches and stresses within the two isolated constituents. So you can see here, this is a schematic for each constituent. So this psi here could assume the value M or C to describe myocytes and collagen respectively. And you can see we define a mapping between the cut and nearly stress-free configuration for each constituent to the uncut traction-free configuration for each constituent. This is the type of deformation gradient that we're employing here, and which is a function of course, the geometry in the open and in the closed configuration, as well as the opening angle. And then we also can, because we're assuming compressibility for most of these deformations, we can also apply volume conservation to be able to connect and map each value of the radius in the open configuration to each value of the radius in the closed configuration. So as I said, the experiments are used both to validate, but also to inform this model. And in this description, the value that we're taking from the experiments are the inner and outer radiuses of cut samples, both for both isolated constituents values, which are represented here with this capital RI, as well as the opening angle. But we also have two unknowns that are left in this deformation. And so we need two equations to be able to evaluate those. And these equations are, of course, the equilibrium equations, both in the radial as well as in the axial direction, which we can use to solve for the inner radius in the closed configuration, as well as the axial stretch in the closed configuration. And here again is what we're taking from the experiments. Now that we know everything in the closed configuration for each constituent, now we can put the constituents together. And this is where we are introducing this FC interaction and FM interaction, which accounts for the mechanical interactions between the two constituents. Here you can see how we have decided to shape this interaction using this deformation gradient here. And again, we can use this radial mapping between the constituent-specific uncut traction-free configuration to the same before the mixture. We have, again, two unknowns here, which is the inner radius in the uncut configuration for the mixture, as well as the axial stretch in there. And we also have this parameter here. I just want to point it out. We'll discuss it more later, but this is actually the parameter we're using to modulate the mechanic and describe the mechanical interaction. We have decided to use this kind of isotropic swelling-like type of shape because we'll discuss more later. So again, we have two unknowns, so we need two equilibrium equations, which are now the radial and axial equilibrium equation in the uncut traction-free configuration for the mixture. And again, here down here, there's a summary of what are our variables. And so this is what we're getting from the experiment, which are the same as the described before, plus the composition of the tissue. As we said, it's 10% collagen, 90% myocytes. But also now there's some of the geometrical quantities have been estimated in the prior step and are used now as known variables in this putting together the two constituents. And we're solving again for the inner radius and the axial stretch in the uncut traction-free configuration for the mixture. Finally, the next step is to apply a cut and open the mixture, the intact tissue configuration. And so we use a similar structure, but this time we have three unknowns, not just the inner radius and the axial stretch in the open cut equilibrated configuration for the mixture, but also the opening angle. And so we need three equilibrium equations. We can add a circumferential equilibrium equation that you can see written here. And again, this is a summary of what are the known and unknowns of this type of problem. So again, just a reminder, alpha C and alpha M is what we're using to modulate the interaction, mechanical interaction between the two constituents. Here's a little bit of the workflow, how this model, how we have employed this model. So we start from the inputs, which are the experimental values for the material parameters, as well as for some of the geometrical parameters. And then we set a set value for alpha C and alpha M, which represents the interaction. We then solve for all the equilibriums that you have seen through the kinematics, and we obtain as an output of the model the predictive value of the opening angle of the intact tissue. Then as a validation, we can compare it with the experimental value that we measure for the same tissue. Now, a question is, do they match? Well, if they do match, that means the inputs and especially the interaction parameters are a solution of the problem. And so this solution predicts accurately what we've seen in the experiments. If the two values don't match, then that means the inputs are not a solution for the problem. And so we need to try a different set of parameters. So we'll use this flowchart to infer if alpha C and alpha M matter. So first question, do we have to consider them? And the second question is, if we have to consider them, what shape can we use of this interaction to understand how collagen and myocytes work together? So I'm starting with the model prediction without any interaction, just to orient yourself here in the horizontal axis. We see the values of the collagen mass fraction. That means when this horizontal axis is equal to 0, we have a isolated myocytes condition. When it's equal to 100%, we have a isolated collagen condition. The experimental intact tissue is represented by this vertical line here for a 10% collagen, 90% myocytes. On the vertical axis, we have the predicted opening angle, and you can see here the three values that we have also found from experiments, which is the myocytes value of the opening angle here at the bottom, the intact tissue here at the center, and the collagen up here. This green line here represents the prediction of the opening angle for a varying amount of collagen within the tissue for when there's no interaction. So when both alpha M and alpha C are equal to 1. So this gold star is what you need to keep your eyes on, because this represents the experimental condition. So tissue composition, 10% collagen and 90% myocytes, and recorded opening angle of 58%. Every prediction of the model that passes through that gold star means it's a solution. Every prediction of the model that does not pass through that gold star, it's not a solution. First, we were kind of happy because we have seen that even without interaction, our model fits the boundaries that we expect. So for complete myocytes, for isolated myocytes, it recovers the value of the opening angle for the myocytes, and for isolated collagen, it recovers the value of the opening angle for isolated collagen. However, when we look for the actual in vivo kind of composition of the tissue, we found that the model without interaction does not passes through the gold star. Actually, it predicts a much higher value of the opening angle for this tissue composition. And also for the model to be able to predict the correct opening angle, one must use a composition of the tissue that it's 0.06% of collagen and the rest myocytes, which is not the composition supported by the experiment as we have seen. So this made us think that the model without interaction seems to be unable to recover the experimental results. So the next step was for us to try and see what is the role that this interaction parameter can play within this model. And so here we started by considering the interaction parameter for the collagen to be smaller than 1 that represents a compression for the collagen network, which seems reasonable since many images of the left ventricle show that the collagen is in a wavy configuration when in the tissue. And then, but we decided to keep the myocytes undeformed for this first series of results. You can see here the effect of alpha C is that the increase in alpha C makes the opening angle increase, or really more the decrease. So increased compression decreases the opening angle in the intact tissue. And the value of alpha C equal to 0.94 makes it so the prediction passes through the gold star, so that that can be considered a solution of this problem. This only represents the first 10% of the horizontal axis because all of these curves plateau after that. And so we decided to represent only the first 10%. Then the second type of interaction we tried out was myocytes in tension and collagen in compression. And actually the myocytes is the reverse value for the collagen here. And so we saw that an increase in tension in the myocytes creates an increase in the opening angle when you have lower amount of collagen within the tissue. And an increase in compression, just like before, generates a decrease in the opening angle when you have an increased amount of collagen in the tissue. The solution that passes through the gold star, so that's a solution of our problem, can be found for a value of compression of the collagen of 0.89 and tension in the myocytes of 1.12. We've also tried to see what happens if we apply a compression to the myocytes. And so all of this graph is a little bit different. All of these are solutions of the problem, and we'll discuss more about this in the limitation. But as you can see, all these curves pass through the gold star. So these are all solutions of our problem. And our question was like, okay, is there a solution that that has a compression for the myocytes and tension for the collagen? And what we have seen is that in order to find a solution like that, you have to compress the myocytes really strongly. And so this seems to be potentially not what we observe in vivo. And also it seems to suggest that the collagen is the strongest contributor to this interaction and to the overall residual stress state within the left ventricle. So in summary, what we, in this study, what we have done is to measure for the first time the opening angle for isolated constituents. And we have observed the difference between intact tissue collagen, isolated collagen, and isolated myocytes, suggesting that different constituents can contribute differently to the overall mechanical condition of the tissue. And through the model, we have shown that, we have shown that traditional mixture theory can now recover the experimental measured value of the opening angle. And this model can be used to quantify, or at least to make a hypothesis on how the interaction changes it could be used to understand how the interaction between collagen and myocytes change in pathological remodeling. There's been another recent study that suggests also the existence of mechanical interaction that you can see cited here. But it's a bit different than what we have done because they infer, first of all, the existence of interactions in the right ventricle, and by not comparing to the residual stress, but comparing to the biaxial mechanical properties. And they've also decided to model this interaction by an additional myocollagen coupling strain energy density rather than using a kinematic model like the one we're proposing here. There are some limitations, of course, to this study. First of all, tissue treatment. There's been reports that the desolarization and the collagenase treatment we're using have not affected other tissues in the past, but we're not sure 100%. We don't think there's been the tissues have been affected by these treatments that we applied, but it's a limitation because we didn't have a good way to actually quantify the fact that the collagen and the myocytes were not affected mechanically. And also another limitation is that it's hard to estimate if the myocytes are completely in the passive behavior. And we have treated the tissue with BDM when we had the presence of cells. And when comparing our intact and isolated myocytes opening angle with the opening angle reported before of a fully contracted left ventricle, our value is much lower, which seems to suggest that at least there wasn't like a strong contraction in our tissue. But again, we haven't quantified that, so there's a limitation. We're also using an isotropic description for both constitutive behavior and introduction and interaction, which is a limitation. We know the left ventricle, it's not isotropic, has a microstructure with that as directionality. This was a first attempt, and we didn't have a lot of information about the specific distribution within our tissue. And also we weren't able at the time to estimate the mechanical behavior of the actual tissue that we had. And so to us, this was the first step, and but we recognize this is a limitation. And finally, another big limitation is that our results are not unique. As you have seen, I've shown you multiple solutions of the problem for multiple shapes and parameters combinations. And this is a big deal because that means we are postulating there is an interaction. Our results suggest that there is an interaction, but we cannot really make any comment about the specific shape of this interaction. We need a more sophisticated model and more sophisticated experimental measure to be able to inform the shape of the kinematics of the interaction. And in conclusion, this is, I think, what this paper was trying to prove is that cells and extracellular matrix can exchange forces in the left ventricle, and it is important to account for these forces when modeling the mechanics of the left ventricle. And we have proposed an innovative model to account for these interactions, but this is not perfect, and we need more experimental data and a more sophisticated model. As a very last thing, I want to say this is relevant to new emerging heart failure treatments, as well as regenerative medicine, people who are thinking about designing scaffolds that are hospitable for cells. I think it's important to keep in mind the mechanics of the scaffold. Cells really strongly respond to mechanic, and this could be a way of also help inform this type of design. And finally, I want to thank all the students in my lab, and especially Marisa Grobel, who is the woman behind most of these results. And also I want to thank all of the collaborators that contributed to this study. And thank you for being here today, and I'll be happy to answer any question.
Yeah, thank you so much for your talk. Yeah. Yeah, please, people who have questions, either feel free to speak up or participate in the chat.
Hi, hello. Hi, Sara. Great talk. This was very interesting. It actually answers a lot of things I was wondering about myocardial tissue in the past as well. And I see how difficult it is to even to start answering some of these here. Just one thing I was wondering, you mentioned that it's difficult to get like a unique solution to these interactions. But I'm not an experimentalist, but I was wondering, like, is there some way that you could work in the deceleration process that you have different amounts of collagen fraction still left in the experiment, so you have more data to sort of see what the interactions could be?
This is very interesting. This is something that we have been thinking about. It is hard to estimate specifically because the decellularization, it's more like an on and off kind of process. So the cells are either lysate or not. And the collagenase, so I would more be inclined to look at a collagenase procedure. And it is hard to estimate exactly what you're doing to the collagen with the collagenase procedure. But I think this is a great idea. That's one of the directions that we're thinking about going, to try to have more of a gradual amount, to try and define what is the shape that the opening angle is taking as varying the change of the amount of collagen. And another thing we have been working on is to use more imaging on the same tissue before and after one of the treatments, to try to actually inform a actual mapping of the deformation, what it looks like. And it's hard to find landmarks in the tissue that can allow you to kind of like map back, but this is one of the things we've been trying and thinking about. And another thing that it's important for us is to have more information about the microstructure in general, organization in the actual samples, and so more coupled information, which takes away a little bit from the generality of the model, but it can help inform a specific shape of the interaction. And yeah, but this is a very good question. Thank you.
I know. Sara, I have a couple of questions. We don't do so much LV work, but RV work, and in essence, I'm sure some people will disagree, but they don't fundamentally differ in their sort of microstructural makeup. And we went through a lot of pains to recently test the myocardium under all kinds of deformation modes, with the thought that it's such a complex orthotropic material that you really want to elicit modes where, for example, that shearing and that resistance to shear that may be transferred through interactions of collagen, the extracellular matrix, is being accounted for. I wonder, you haven't done sort of mechanical analyses or experiments beyond the pre-strain experiments, at least in this paper, right? So how do you envision what you found now will translate to sort of the broader scope of myocardial mechanics?
Yeah, 100%. So right now we use the opening angle as this was kind of like a proof of concept first paper to try to understand. But I agree that I think what we really need to be looking more into is there are mechanical ways that you can measure the interaction between the two. Like you said, for example, using more of a shearing type of deformation, or and so I think this is the direction that we need to go, to have like a more specific mechanical quantification. And I also think that, for example, having pathological models, so a pathological way of changing the amount of collagen, or the amount of the behavior of the myocytes, or a more pharmacological way. So I think also going and looking into the active contraction could help in inform all of this. So I think in general, we just need more information. It can be in a more sophisticated way to measure the passive behavior. It could be in a more sophisticated way to measure the active interaction, or using the help of a pathology that we know we can estimate how it changes and use it as kind of like a benchmark to estimate how our predicted results will account for this change in composition and microstructure. So I think this is a direction. The idea behind this was I was thinking a lot about stress-mediated remodeling, and I really think, and not that by any, not by any measure this just answers all the questions, but this is the direction we were thinking if we want to improve the stress-mediated growth and remodeling models, we really need to be thinking, how do we connect this more in vitro series of experiments that are available on isolated cells with the in vivo condition? And this does not only include the configuration, but also, I think, the interaction with the rest of the constituents in the tissue. I don't know if this answered your question.
It does 100%. There's so many other questions, but I want to give space to people that also have questions. I don't want to hog all the time. So please, if you have other questions, make me shut up.
I have a related question. So do you know if anyone's done any opening angle experiments on like pressure overloaded ventricles? Because I know that, you know, there's a lot of fibrosis that can happen after like those types of pathologies.
Yes, so yes, Fang have done opening angle on everything you can think of, including a hypertensive remodel left ventricle. We also have recently published a paper that shows opening angle on a hypertensive model of doll S animals. And we do see a change, although one of the limitations of the paper that, one of the limitations of what's out there in the literature is that it's kind of like a ligature type of hypertensive model. And so that has a lot of inflammation that comes with the surgery within itself. So it's kind of hard to parse out what is due to the increase in pressure and what is due to the inflammatory response, in my opinion. The model we've used is more of like a genetic model of hypertension through a doll S. But in that case, it's kind of hard to make sure you're using the correct control. And so, you know, no model is perfect. We're all trying to do our best. But definitely there is a change associated with hypertension, which affects actually different constituents. And so that we thought was interesting. We're working on applying this model on those. Kind of 2020 throw us for a loop there, but hopefully 2021 will be better. But yes, there is an opening angle in hypertensive, yeah.
And I also had a second question, if that's okay. So I really love that you did both male and female animals. And I was curious, what do you think is contributing to the differences in the opening angle?
That's a good question. So the opening angle, actually, look at this. So I think the opening angle, it's a good measure in my opinion, but it's still somewhat of a dimensional measure. So it's not non-dimensionalized.
Sure, yeah.
Yeah, and so I think part of it could be a little bit of a size effect. That's as far as I got, but this is something that we're really interested in. There's really not a statistical difference in none of this, neither collagen or intact tissue. We did see a difference in the myocytes, and the difference is actually, there's more differences when you look at a hypertensive model. I actually think I may have some results here. If you're interested, maybe I can show you later. But so yes, I don't know, but I think it's really important to look into it, especially when you look at remodeling, because there are, you know, sex differences that are observed on how the development of hypertensive pathology. And so I think it's important to look more into it. Yeah. Okay.
Yeah, that's awesome. I'm looking forward to that study, because that was one thing I thought about too, is that, you know, female animals usually have less fibrosis during pathology than male, which I always thought was really interesting. But it'd be cool to see like what the mechanical, you know, consequences of that would be.
Right.
That's awesome.
Yeah, I agree. And I think it's still an open question. I don't think there's a lot of clarity about it. And I'm not telling you that this is the answer, but I think it's one of the avenues we can go and look into, at least to gain more information about what may be contributing to these differences, right? Because it's interesting.
Yeah, it's super interesting. Thanks.
No problem.
Yeah, I think we have time for maybe like one more question if someone wants to ask one. Kyoko, I was going to ask your second question.
I have a somewhat maybe provocative question, but
Okay.
given that your work highlights the sort of complex interaction between constituents, do you think there is space for constrained mixture models for complex heterogeneous materials like myocardium? Do you think constrained mixture models can capture these effects and accurately represent the constitutive behavior?
So that's a good question. The answer is I don't know. Because I really feel about two minds about it. I really enjoy the simplicity of it and of constrained mixture models, and also kind of like to a certain extent the computational cheapness of them when you compare them to a fully microstructural model, right? But I think it's still a question if they have this ability. I think we can make them better. I think thinking about this outside the box about this type of interactions, as well as even more like shape effects, and that there's ways that one can, I believe, can include those. But I just don't think I don't have an answer to say I don't think there's a perfect model. I think every model has pros and cons. And I think there are still enough pros in the constrained mixture models that I wouldn't just like discard them completely. But I definitely see the limitations, and this is kind of like the direction this project was going in, trying to address, still remaining in this philosophy of being like simple to, and kind of like assign a clear role to the parameters that you are inputting, so that you know what mechanically you are modeling, so you can manipulate it and interpret it a bit better. But I also see a lot of pros in fully microstructural models, and if that's what you're thinking when you're saying comparing to, I don't know.
I wasn't necessarily thinking. I mean, we've recently done again pretty extensive experiments on RV myocardium, and we fit it to a constrained mixture model. And you come up with some measures that may be interpreted in microstructural ways, but right, if they're fundamentally flawed, if the idea that your constituents are governed by one shared deformation is just fundamentally wrong, then your interpretations naturally are also wrong. And I think we as biomechanicians have been very focused on these constrained mixture models, right? Of course, there's microstructural models, but sort of on the organ and tissue level, they're generally not very useful. But other disciplines use other forms of material models, like shear lag modeling, for example, that I wonder whether your data seems to nudge us in the direction of at least exploring more the limitations of the models we use. That's just sort of a thought I had as I listened to you.
Yeah, no, I agree. I think we are the new generation, right? So maybe that's that's the direction we should be thinking. I don't know. I definitely share the fact that I share I like these models a lot, and these are like the models that I've been working with, until now. And I think I built an expertise to understand these models to a certain extent, but I am, as you can tell, I'm also thinking how can we make them better? And I have not hit a wall yet, but I'm not excluding the possibility that will happen in the future. But I think when you're trying, I think there's still power, as I said, like when you're trying to quantify, I think they still may be helpful in the sense of, in a comparative kind of sense, like when you're trying to quantify the effect of a pathology, quantify the effect of a manipulation, and you are not really looking to connect it to a microstructural necessarily behavior. These models can still give you because it's a very limited number of parameters, I can still give you information, but it's a trade.
Awesome. Thank you, Sara. I appreciate it.
Yeah, it might be good to end on this sort of philosophical note, unless anyone has like a dire burning question. Yeah. Well, I'll stop recording now. Thank you.